Wonderful. So thank you very much. Thank you everyone for joining today's lecture, and it's my very special pleasure to introduce today's speaker, Dr. Elizaveta Gorodetska. I know Lisa many years already, and Lisa now is postdoctoral fellow, a very well-established scientist at the Campoid Center of Dresden-Rosendorf. However, when we first meet many years ago, I believe that we met at the Taras Shevchenko National University of Kyiv, Ukraine, and we discussed the future strategy, how Lisa could potentially join our group. It was a very touching moment. And I think a couple of years after our meeting, Lisa joined our group through the program for, I believe, German Academic Exchange Service, right, Lisa?
Yes, yes.
So it was a DAD grant, and Lisa got an opportunity to join our group and to start her PhD. And well, a few years later, she successfully defended her doctoral thesis, very successful. So she received also an award for her doctoral thesis, and now she is principal investigator for many exciting projects, including a project which was awarded recently by the large grant and focused on liquid biopsy. But the major focus, scientific focus for this research is understanding and analysis of the role of cancer stem cells in radioresistance and how this population can be targeted. And this, I believe, nicely overlaps with today's topic of her lecture. And today, Lisa will explain you how radiotherapy kills tumor cells, and she will also touch 6 R's of radiobiology and also explain why cancer stem cells may contribute to radio resistance in the light of these 6 R's of radiobiology, and how biological 6 R's can be applied in clinical practice. So we are very much looking forward for your talk, Lisa. Floor is yours.
Thank you so much, Anna. So I will now try, I tried it already, but to share my screen again. Yeah, so do you see it in presenter or presentation mode?
Not yet, not in presentation mode yet.
And now?
Yes.
Yes.
Okay, perfect. So then I will just choose the pointer and can basically start. So yeah, thank you so much for having me here and for the nice introduction. Yes, as was mentioned, with a lot of spoilers, we will talk today about how does radiotherapy kill tumor cells. So I will start with the agenda basically for today. So I will start with a short introduction into the topic. We will discuss ionizing radiation first. So what is that and why it's ionizing? Because it's really important for the killing, the initial or end point. We will then touch direct and indirect effects of irradiation, following by 5, 6, or 7 R's. We will have to figure it out today together with you. We will, of course, talk about cancer stem cells and about tissue and cellular responses. So I will jump immediately into the first part, which is the introduction. So it's important to define what is radiotherapy. Yeah, so radiotherapy or radiation therapy is a type of cancer treatment, not only cancer, but mostly, that uses high doses of radiation to kill cancer cells and shrink the tumor. It's known by statistics that around 50% of all cancer patients will receive radiotherapy eventually during the course of the disease. And this is a very huge number, you can imagine. So it's a very popular method, which is widely used all over the world. And in radiation therapy, ionizing radiation is used for the therapeutic purposes. So not just some kind of irradiation, but you will understand later why I have here the water bottle next to it. So starting with the types of ionizing radiation, that I picked this cartoon to show you or to introduce you to the two main ways to use radiation, but also to the types. So starting from the types of radiotherapy can be used in very different ways, mostly in two ways, which is external. This is the most common and most available, which is external beam radiation therapy, or EBRT shortly. And this means that basically the machine directs beams at high energy radiation towards the tumor. So the patient is being put into this machine, and the machine is rotating around, and or not rotating, it depends. And the beam is delivered. So it can be the X-rays, electrons, or protons, whatsoever. We will touch this a little bit later. The next type of radiation therapy, it's internal radiation therapy, which means that basically that radiation source, some kind of like beads, are put inside the body, so in the close proximity to the tumor, or systemic therapy. And this radioactive material then finds the cancer cells and destroys it. It's also, so can be, for example, for prostate cancer, the brachytherapy is used. So these are the two most widely used types of radiation therapy in the clinics. But now coming to the ionizing radiation, which is actually used with the therapeutic purposes, I have to mention that ionizing radiation uses the high energy beams, such as X-rays, so photons, or gamma rays, protons, or electrons, to damage the DNA of cancer cells. And this leads to the killing them or losing their ability to divide. So it's important, what I wanted to show on this picture, how different the photons and protons are. So you can see here that the photons, or X-rays, they have no mass, and they deposit their energy along the entire path. So the photon beam travels through the body, potentially affecting also healthy tissue, not only the tumor, but surrounding healthy tissue, before and after the tumor location. The protons instead are charged particles, and they can stop at the specific depth. This can be, so to say, regulated by the phenomena or the fact called Bragg peak. I will also talk about it a little bit later. So this allows the protons to deliver the high doses of radiation directed to the tumor with sparing of the normal tissue. So this is really beneficial for the patients. And in the real life, this is how it would look like. So you can see here the photons or X-rays and protons, and the beam scan of the brain. And you can see how the dose is distributed in one case and another. Touching the Bragg peak, which I just mentioned before for the protons, it's really important to understand that the Bragg peak is a specialized, highly precise, and localized peak of radiation dose in proton therapy. And it allows the oncologist to deliver the maximum of energy to the tumor and causing the minimal damage to the normal tissues. You can see on this graph that on the x-axis it's the depth or distance inside the materials, or in the body in this case, and on the other axis you can see the dose. And basically when we apply it, we can see how different they are for the X-rays, where we cannot control. So it can be the X-ray treatment, the dose can go through the normal tissue as well, and the proton is really specific. And I also would like to mention here the person who discovered it and described it. It was oh, it's not working. Oh, working again. So it was British physicist William Henry Bragg, because of whom, it was later named the Bragg peak. And he discovered this phenomena, but he was also receiving the Nobel Prize in Physics in 1915, but for the other thing, because he was radiation physicist, he was working a lot with the X-rays as well. So he, together with his son, they received shared Nobel Prize for the services in the analysis of crystal structure of the X-rays. So now about basically more how it looks on the cell level. So you saw now brain scan, you saw some graphs from the physics point of view. And on this slide, I wanted to show you the example of the real mammalian cell and how protons or X-rays can actually affect the nuclei and cause the damage. So the blue is the nuclei of the cell, and the green is the so-called gamma H2X. It's a marker of the DNA double-strand breaks. I will talk about this a little bit later. But what you can immediately notice here, without knowing actually what you are looking at, is that the protons are really going through the specific line, and the photons are more distributed. Important that doesn't matter what radiation we apply, it leads to the DNA damage. And what I have to mention also here is that you will see these QR codes through the whole slides I prepared. You can just simply scan them and follow the papers where I took the info from. So in case you want more insight, you can use it for later. So now, since we talk a lot about the types of radiation therapy and what damage it can cause and how we can apply it, I would come to the first quiz question, which I prepared. So I don't know, Natalia, how it works. Do I have to click something?
No, I will launch it. So I just launched it.
Okay, yes.
You can read the question, so when you talk, it's also
so the
subtitled.
the question is, what is the primary difference in energy deposition between photons and proton beams for treating a tumor? And you have 5 options, and only one is correct. So we will wait until we are 80% or will answer.
Okay, so we have 83, 5, 7%. So ending the poll, sharing the result.
Yes.
So majority answered C, 65% answered C.
Yes, very good. So C is the correct answer, that the protons are stopping inside the tumor and we basically can control it. Yes, so I can go further.
Yeah.
Okay.
You can close it.
Yeah, I think I did.
Okay, great.
Yes. So I'm happy you got the information correct from me. So I wanted to also share some small take-home message from this part. So there are basically 2 main types of radiotherapy, external and internal. Photons and protons are fundamentally different types of radiotherapy. And important to remember that the Bragg peak is a localized maximum where the radiation dose deposited by charged particles like protons. And in the end, the tumor cells die because of DNA damage. So which you saw on my last slide, which leads to the cell death. So now coming more specifically to the cell level, the next part of my talk will be about direct and indirect effect of radiation and what does it has to do with water, actually. So many people are getting surprised with that. So if you want, again, more info, you can scan the code or follow the link when you will get the slides. So I'm happy to share them also. And we know that our body consists of water, at least at 60%, and this is what we are being all told at school, and so we know that as just a fact. When we also look at the cellular level, we can see that the cells, which is the basically a building block of a living thing, is also consists mostly of water. So around 70% of the cell is water, and only 30% is I don't know, hard part. So chemicals. We also have to understand why radiation kills and how radiation kills the cells. We have to look really deep into the cell. So if the cell is basically a building block, inside it there are different organelles, so different parts of it. And one of the important part is the nucleus. You can think of the nucleus as I don't know, brain or a hard drive, basically, where all the information is located. So it contains the chromosomes, and they are made out of basically DNA. But DNA is not just floating around, it's just loosely packed there into this structures called chromosomes. So why we have to look on the DNA level, I will tell you now immediately in the next slides. So when we apply radiation, ionizing radiation, it can cause direct effect by directly causing the double or single strand breaks, leading to the cell death, or indirect effect. And this direct effect is direct ionization of the DNA. And the indirect effect, which is the most common for the X-rays at least, is the production of free radicals. So in any case of indirect or direct damage, we will have unrepairable DNA damage, which will lead to the cell death. So now talking about water, what it has to do with water is that, as I mentioned before, the cell consists on the 70% from water. Yeah. So, and in the case of indirect damage, the radiation interacts with the water molecules inside the cell, leading to the radiolysis of water. And radiolysis of water is the composition of water molecules into highly reactive free radicals, some hydrated electrons and hydrogen atoms, or molecular products like molecular hydrogen or hydrogen peroxide. And those things will actually damage the DNA inside the cell. I found very nice video lesson about how it works. It was really nice graphs. So if you want, you can really check it. It's really beautifully made. So, okay, the DNA is damaged, but what kind of DNA damage this is? And for this, we have to look deeper again at the DNA. So DNA is, I think most of people know, it's desoxyribonucleic acid, which contains containing the instructions. As I said to you, you can imagine it's a hard drive with very important data. So it contains the genetic instructions for the development, functioning, growth, reproduction of the organism. When we look at the DNA, we can notice immediately that it's a double helix. So it's like a ladder kind of thin structure consisting of two anti-parallel polynucleotide strands. And the backbone of it is the alternative sugar and the phosphate groups. And attached to this sugar is one of the bases, which you can see nicely here. So why we are looking at this? Because the damage which irradiation can cause in DNA is very different. And there are basically 4 different types of DNA radiation-induced lesions, which were described. And they are the base and sugar damage, which is basically modification of one of the 4 DNA bases, which I mentioned previously. Then there are single strand breaks. So single strand break is just break of one of the sugar phosphate backbones, so one of the part of the ladder. And there are double strand breaks. Double strand breaks are basically the break on both complementary strands, usually in a very close proximity, which causes to break the DNA completely. And there is also cluster damage, which is basically the region containing 2 or more lesions, so different damages. It's important for the understanding what actually happens. So we know already that DNA double-strand breaks are the most lethal. So when the DNA broken completely, this in most cases leads to the death. So now also I wanted to mention, which I think is really important for the biologists, but also for the clinicians, is how to detect this damage. And the most, I don't know, popular or very much established in the biology way to detect the DNA damage in the cell is the gamma H2AX. It's this blue cells, which I showed you in one of my previous slides. So what is that? It's basically a highly sensitive early molecular marker of the DNA double-strand breaks and the replication stress also. It basically recruits, it comes one of the first to the point of broken DNA and just like, screams there. So it recruits other DNA repair proteins to the site of the damage, and we are using it very widely in our research. So you can see on this picture, again, the nuclei in blue, so the DNA, and there is damage on it, which is this green dots, it's this gamma H2X. Gamma H2X, so H2X is the histone, one of the histones, and it's a phosphorylated form of it. And yeah, as I already mentioned, it serves as a marker of DNA double-strand breaks. So now we are coming to the next question. Please, Natalia, help me. So in the context of ionized radiation, which DNA lesion is characterized by most irreversible genomic instability or cell death? And very similar question was yesterday at Leonie's lecture. And I first wanted to change it, but then I thought, okay, we can see how the repetition helps to remember information. So yeah, we will wait until people vote and I will comment.
But I think people remember the lecture was just yesterday.
Well, I also don't know if it's exactly the same people today here or not.
Mhm. Okay, we're almost there. A few more answers, please. Excellent. So I'm ending the poll, sharing
Yes.
results. So 77% answered D, double-strand break.
Yeah, so very good. It's true, it's DNA double-strand breaks. I think the people who didn't respond correctly probably were not yesterday at the lecture, because it was really nice introduction to the basics of tumor cells. So yes, it's true, the DNA double-strand breaks are the most lethal, and we will talk about them more in the next slides. So the take-home message of this part is that ionizing radiation damages cells through the direct interaction with DNA or indirectly via water radiolysis. So this is what it has to do with water. Indirect damage includes DNA alterations like double, base, single, and cluster damage. And double-strand breaks are very often called in the literature the lethal lesion, because they are leading to the cell death. Now coming basically to the core of this lecture, is that I have to start from the clinical point of view that when we apply radiation, we usually don't apply it as a whole big dose simultaneously, but we apply it in fractions. And for this, there is a very rational explanation, biological explanation, which I will guide you through today. So first, the rationale for this fractionation was summarized as the 4 R's of radiotherapy by Austrian scientist Hubert Rodney Withers in 1975. So you can find actually this paper was published in this journal and then it was scanned. It's very bad quality, but you can find it, and actually it's very interesting to see how the paper looked in 1975. It's 2 pages, but it's a very good journal at that time. So you can check the QR code again. Yes, and in his paper, he basically wrote that ionization caused by radiation are distributed randomly in matter, and hence cells killing is a question of probabilities. So a dose that inactivates half the cell population will, if repeated, inactivate half of the survivors. So it's that I highlighted the key points. So basically, yeah, repeating those dose in fractions makes sense. And what he offered us, so he offered so-called 4 R's of radiobiology, yeah, or radiotherapy. It's the fundamental biological principles, and they consist of the repair, which means DNA repair, reoxygenation, redistribution, and how he called it regeneration, but later it was renamed to repopulation. So we'll now go through all of them. I will try to explain you what is their meaning and why they are so important. So the first R of radiobiology is the repair. You already saw on the previous slide the DNA double-strand breaks, our little lesions, and repair is one of the primary reasons for fractionated therapy. It's because that when splitting the radiation dose into small fractions, the cells are having time to repair the sublethal damage, especially the normal cells here, of course, meant, yeah. So they can have time to repair themselves, and hopefully the cancer cells will die. So normal cells are in general better at repairing. That's why it makes sense to do the fractionation. So the tumor cells often have deficient repair pathways. So we also, of course, to have mentioned the repair pathways. I don't want you to remember all the proteins involved, but it's important to know that there are different pathways or different ways how the cell can repair itself. It doesn't matter if it's a normal cell or it's a cancer cell. So the DNA, the two main mechanisms how cell can repair the DNA double-strand breaks involve non-homologous unjoining and homologous recombination. And non-homologous unjoining is the dominant, rapid, generally error-prone pathway. So it can make also errors and troubles to the cell, but it repairs it in the end. Homologous recombination is more slow. It's really highly efficient and accurate. And it uses the homologous template, so the sister chromatid to repair the damage very accurately. And I put here like a table with several main differences. So as mentioned already, the non-homologous unjoining is the error-prone, homologous recombination is error-free, and non-homologous unjoining is direct ligation, which means that there is a break, we just clean it and glue it together, job done. Homologous recombination requires the sister chromatid or homologous sequence. Non-homologous unjoining is independent of cell cycle, so it doesn't matter in which stage cell is, we can repair it. Homologous recombination in S and G2, in the phases where there is a sister chromatid. And non-homologous unjoining is super fast. Homologous recombination is really slow, and yeah, non-homologous unjoining can introduce the insertions, deletions, so to make errors on the DNA. Homologous recombination is very precise. So that was the first R, the repair. The second R is reoxygenation. What is meant is the process where the hypoxic, so the cells which lack oxygen, tumor cells become oxygenated as tumor shrinks following radiation. What is meant? So the bulk tumor has so-called hypoxic core, so there are cells inside the tumor which have less oxygen because they are further from the vessels and so on. When we apply radiation, we kill the cells which has a lot of oxygen, and we stay with the cells which are hypoxic, so the radiation is not able to kill them directly because hypoxic cells are very radiation resistant. So we wait until the next fraction, until the cells, the one which were hypoxic, reoxygenate themselves. We apply the fraction, and this way we can shrink tumor, till the end. So oxygenated cells, as mentioned already, 2-3 times more sensitive to radiation damage. and radiation kills these oxygenated cells, and this leads to the overall reduction in tumor size. Then oxygen reaches the edges and it goes in circles again and again. So reoxygenation is very important. When talking about reoxygenation and oxygen in general, we have to mention the oxygen enhancement ratio. This is basically the ratio of radiation dose required to cause a specific biological effect in the hypoxic, so low oxygen conditions, compared to the normal oxygen conditions. In radiotherapy, basically measures how much radiation is required to kill cells in hypoxic conditions. So you can see on this graph that aerobic cells versus anoxic cells, how different their responses to irradiation upon the condition of the oxygen in which they are located. For the reoxygenation, I thought it also makes sense from the clinical point of view. Also mentioned that with oxygen, there were attempts to make cells more radiosensitive, so more sensitive to the radiation. And for this, so-called hypoxic radiosensitizers were established. Those are the agents which are designed to enhance the killing of the radioresistant low oxic cells within the tumor during radiotherapy without actually influencing the normal cells. They usually act as electronophoenic compounds and substituting for oxygen to make these cells more vulnerable for radiation-induced damage. So there are some key agents like nimerazole and so on. And there was also, I think, Professor Krause in her first lecture mentioned this trial. It was a very famous, big trial on the head and neck cancer, where you can check this online also, where it was significantly, the survival of the patients was significantly improved when using these radiosensitizers. However, in the recent years, it shows it doesn't work well for all the cancers, but in head and neck cancer, it worked. So you can see the blue is radiotherapy only, red was the hypoxic radiosensitizer, so it is getting better. So the third R of radiation biology or radiation therapy is redistribution. With redistribution is basically refers to the movement of the cells through the cell cycle, because of course the cells are not standing on the same place. Yeah, they are dividing and moving through the cell cycle. So yes, so the cellular radiosensitivity is dependent upon the stage in which the cell is now in the cell cycle. What does this mean? That cells display different radiation resistance in different cell cycles, and for example, towards the end of the G1 phase and into the S phase, where the cells in G2 phase are going through the mitosis, then we see bigger radiosensitivity. It's because that, for example, in the S phase, cells just completed the DNA replication or just finishing it, and they have sister chromatids, so it's more hard to affect them because it's two chromatids together, so they are more resistant. And so on with the other stages of the cell cycle. So this is also important to keep in mind, because again, fractionation gives us time to catch cells in these more sensitive stages, killing them, and then wait until they will go to the next stage and then irradiate again. So that makes perfect sense for the fractionation. So the fourth R offered by Withers is the repopulation. And the repopulation refers basically to the rapid proliferation of the survival cells during fractionation radiotherapy. So the fractionation allows the surviving cells, the normal cells, to multiply, regenerate, and repopulate the tissue between the sessions. And the cancer cells often have accelerated repopulation, so they divide faster the original doubling time, and during the treatment breaks, this reduces the effectiveness. So repopulation is also very important. So now, when I mentioned all 4 Rs of radiobiology, which I will remind you is repair, reoxygenation, redistribution, and repopulation, I would like to come to the next question. And the next question is, which of the following describes the radiobiological rationale for the fractionated radiotherapy based on the force R? So again, there is only one correct answer. Very heterogeneous answers we have. Well, but the topic is also not very, not the easiest one, let's say so.
Okay, so we have 83% answering, amending the poll, sharing results. So majority 43% answered E. It allows hypoxic tumor regions to reoxygenate.
Yes, very good. That's correct. I mean, I on purpose put this answers also maybe very not very straightforward, so that you have to be really cautious when you read it, but it's true. The 4R and what was meant here, what is describes the radiobiological rationale is that yes, it allows hypoxic tumor regions to reoxygenate. Okay. So I again have the take-home message from you. So what we have to remember that fractionation is a biological strategy. Yeah, it's not just physical one. It basically buys time for the normal tissues to repair, reoxygenate, redistribute, and repopulate, hoping that this will make the tumor cells more sensitive to the next dose of radiation and aims to destroy the tumor faster that it can rebuild itself. However, I have to maybe disappoint you. On the 4R, we are not ended. There was a spoiler already that there will be 6R. So the 5th R was offered later in 1989. Was introduced by 3 scientists, Steele, McMillan, and Peacock, and they published the paper, yes, this letter to the editor, the 5 Rs of radiobiology. And what they actually noticed, this is the figure from the paper. I love the paper. There are 2 figures. I'm like, what a life is that, no? So what they did actually experiment on the cell lines, which was not so much done before with the radiation. So they argued there that these original 4 factors, which were introduced before by Withers, could not explain everything. So they noticed when they began to measure in the surviving fraction of the cells upon different doses of radiation of different cell lines from different tumor entities, they noticed a very wide range of responses. This is what we call now heterogeneity. That we know now that all tumors have to be treated individually. We cannot pool, or not all of them pool together. They didn't know that yet. So what they offered, they said, okay, some cells were naturally tougher against the radiation on the molecular level. So they offered the 5th R. And the 5th R in this case, we know again, repair, reoxygenation, redistribution, and repopulation. And the 5th R is radiosensitivity. What they mean with that is that this baseline radiosensitivity or radio resistance of the specific cells is really different. That's why the response of radiotherapy can be also different, completely logical to my point of view. So what I wanted to show here, are you on this slide, that radiosensitivity defines the intrinsic susceptibility of different cell types and tumors to radiation-induced damage. It can really vary among tumor types, among different cells within the tumor, influencing how they respond to treatment, as seen in the steepness of radiation survival course on the previous slide. What was also noticed later is that radiosensitivity, basically how much damage the radiation causes, is directly proportional to the rate of cell division and inversely proportional to the degree of cell specialization. So basically we have the tissues and cells which are by default are just more resistant and which are more sensitive, and this has to do with how actively they are divided. So this way the fifth R was introduced. But the modern look also says that we are not there yet. And I think this is not the end. There are 6, or some people in the last year say even 7 R's of radiobiology. So recently in 2019, Bostani and the colleagues proposed one more R, the 6th. And this R is the reactivation of the immune system. What they meant in their paper is that when we apply radiotherapy, it stimulates the immune system to recognize and attack tumor cells, often induced by immunogenic cell death. And the radiotherapy regime, the dose per fraction and the consecutive fractions have real impact on the immune regulation. So what radiotherapy does, applied in fractions, in hyperfractionation, in the flash, whatever, it causes tumor cells to release different danger signals and activate, for example, dendritic cells or prime the T cells. And this also makes it different for using fractionation, and also especially when we are talking for the cancers which are treated also with immunotherapy. So the 6R was somehow accepted in the community, which actually didn't happen to the 7th of R because it's very fresh, I think. It's very recent in 2023, so a couple of years ago. The 7th R were proposed, and there were actually several of them proposed. This got the most citations, so I picked this one, which is the reinforcement, that's why R, yeah, of the tumor microenvironment. What is meant? That basically the cancer cell response to radiation can be influenced by its surrounding cellular and non-cellular components, collectively called tumor microenvironment. And this tumor microenvironment can contribute to the survival, progression, metastasis, and resistance to anti-cancer therapies. The major cellular components of the tumor microenvironment are the fibroblasts, so cancer-associated fibroblasts, where they are here, so here, somewhere here, yes. Then myeloid-derived suppressor cells, tumor-associated macrophages, and so on and so forth. And there are also different non-molecular compounds, for example, like lymphovascular networks or extracellular matrix of the cells. And all this together can also influence the radiation therapy response. Again, if you want to check the original paper where it was published, you can get the QR code scanned and used. This, I put here the exclamation mark so that we take it with precaution so far. So we officially like to say we have 6 R's, but there is now ongoing discussion on the 7th R. And as I mentioned, there are several 7th R offered. One of them was related to genetic mutation. I forgot now how it's called, but I can recall it later. So with this, our 6, 5, 6, 7 R's, I'm coming to the next question. Which is that modern radiobiology expanded the classical 4R. Which of the following correctly describes one of these new R's? Very interesting to see how the responses are jumping, how people decide. I can tell you, I was very mean, so on purpose I put only one word start with R, not continuing the whole name of the R, to see how you follow me.
Okay, we have 80% answering, ending the poll, sharing results. So majority said reactivation, B, 42%.
So reactivation is the correct one. So reactivation of the immune response had to be written there, but yeah, as I said on purpose, I just wrote reactivation. So yes, immune cells are really important in the fractionation radiotherapy, in general in radiotherapy. So the take-home message from here, the fifth R, radiosensitivity, represents the intrinsic genetic susceptibility of the cell line or of the cells to lethal damage. The sixth R, the reactivation, which you just responded on the majority pick, of the anti-tumor immune response involves the radiation-induced immunogenic cell death. And the seventh R, which still has to be taken with precaution, the reinforcement of the tumor microenvironment, which means that the microenvironment, so the surrounding of the tumor, acts as a critical determinant of the therapeutic efficacy. And with this, we are actually coming to the efficacy. And we know, so I put here an example of the paper, this example of cancers treated with radiotherapy. As I said at the beginning, around 50% of cancers will be treated eventually with radiotherapy. And there are cancers which are basically cured with radiotherapy, and there are which are not. And for this, there is, of course, a reason. And this reason can be easier to understand through the lens of the cancer stem cells. So while this 6R, which we discussed previously, they provide basically a framework for the tissue response, that cancer stem cells represent the specific subpopulation that often drives treatment failure and tumor recurrence. So in the next slide, we will look deeper into the cancer stem cells and what they are. So I will start from the beginning, from the basically discovery. So this is John Edgar Dick, is a Canadian scientist, very famous one, so the father of cancer stem cells. And in 1994, you can check the original paper here. He and his team were the first to identify and isolate the cells. So they were working with the acute myeloid leukemia, and they saw that these cells, specific for those markers, when to put them into the mice, they can initiate tumors. It was an important discovery, of course. Why it was like that? Because basically the cells, they were able to self-renew. So they had the ability to generate daughter cell retaining the stemness characteristics. And the stem cells also undergo asymmetric division, which is allowing a single cell to divide into the daughter cells with different fates. So basically one can remain a self-renewing stem cell while the other will become differentiated cell. And the differentiation potential, so these cells can restore the heterogeneous cancer cell population, which you can see here. Yes, so the cancer stem cells can then lead to the formation of the secondary tumor metastasis. So there are so-called two main models of cancer stem cell hypothesis. So what are the cells, how to explain them? And the first model, called stochastic model. Stochastic means that all cancer cells have equal ability to initiate tumors, and their evolution really depends on the stochastic influences. So mutations, for example, or some outside kind of noise from the environment. And there is a second one, the hierarchical model, which is explaining that the cancer stem cells are the only cells which are able to initiate, maintain, and seed the tumor. It was thought for a very long time ago. People were arguing. I even remember the times when we had some workshop and people were like almost fighting, like, no, this, what are cancer stem cells, and so on. So it's not very long time ago actually happened. But then at some point, again, John Dick, together with a colleague, Antonia Creso, published this paper, very beautiful paper. I really encourage you to check it if you are into the cancer stem cell or just because it's really interesting. So they said that there is an evolution of this cancer stem cell model. We should not fight with each other, and it's now so-called the unified model of clonal evolution, or also known as hybrid model. So this model basically recognizes that tumors are not just fixed hierarchical or random masses, but dynamic systems. So where basically both genetic mutations and cellular states like stemness can co-evolve. So genomic instability allows multiple cancer stem cell clones to coexist and compete. Basically, we have internal heterogeneity of the cancer stem cells. It's not only that the bulk tumor consists of different populations of cells, but also within the cancer stem cells, we have different populations. So radiotherapy in this case can also act as a selective pressure, favoring the expansion of the clones with better DNA repair kinetics or the antioxidant capacity. And this we have to keep in mind, of course, when applying radiation therapy. But I want to also start kind of the deep diving into cancer stem cells from the where they are located, because they also have a specific place to live. This place called niche. So if the model tells us what the cells are, the niche tells us where they are hiding from the therapies. So this niche can be different, and it usually includes the cancer cells, the stromal cells, the endothelial cells, so fibroblasts, different cells, also exosomes and so on. And there are basically two, the most, so niche is the place where the cancer stems live, so reside. And there are two main niches, which is perivascular and hypoxic niche. And the perivascular niche is localized close to the vessels. That's why it's perivascular. And it supplies cancer cells with the nutrients, so it's easier for them to survive and maintain their status and so on. And the hypoxic niche is one of the critical for the therapy, because it's basically a fortress. It's a region within the tumor, which characterized by extremely low levels of oxygen. And this actually creates physical and chemical, basically, shield against the radiation. That's why the cancer stem cells here are smart enough to reside here, so they cannot be reached by radiation that easily. Sorry, I have to bring something. Yes. And now we know what are cancer stem cells. We know where they are, and we also have to look what functions they have, what they can actually. Yeah, so at their properties. And this is really nice paper. Leonie showed something similar yesterday. It was the hallmarks of cancer in general. And she went very nicely step by step. So I tried to do similar, coincidentally, that I picked these hallmarks of cancer stemness, and also nice paper to check out, which basically defines what are the properties of cancer stem cells. So there are defining hallmarks, which are the self-renewal, for example. So this is the ability to divide while maintaining an undifferentiated state, ensuring a permanent reservoir of tumor-initiating cells. There is also multipotency. So it's the capacity of a single cancer stem cell to differentiate into multiple lineages, so giving rise to the the spectrum of different cells within the tumor. And the tumor initiation, so it's a unique capacity of a single cell, single cancer stem cell, to seed and generate a de novo microscopic tumor. So there are also the emergent hallmarks like differentiation plasticity. This is also a very tricky one. So it's the capacity to give rise to all the diverse heterogeneous cell types found within the original tumor. And that's why it's also very hard to catch them. There are also associate features. So like niche interaction, which meant here that, yeah, cancer stem cells are located at the specific place within the tumor. And the dynamic crosstalk between the cancer stem cells and their surrounding microenvironment provides really protective physical and biochemical signals, so a shield against radiation or whatever therapy. And then also immune escape, so it's the ability to utilize the specialized surface proteins or inhibitory signaling to remain invisible, basically, to the therapy or to the host immune system. And some enabling attributes, which are therapy resistant. So it's the ability to have this very proficient DNA repair, accident fighting, and so on, to be resistant to therapies. And also this ability to undergo the EMT. So this was also mentioned yesterday in the lecture, this epithelial to mesenchymal transition, so changing their phenotype, which allow them to detach from the primary tumor, survive the circulation, come to the organ, make their colony, and start the metastasis. So now, when we know these features, I put them in the context of 5 Rs, or basically it's Anna and some colleagues of us who wrote this really nice paper, which describes the cancer stem cells in the context of 5R at that point, and the general mechanisms of the cancer stem cells radioresistance, as mentioned already before. So it's the enhanced DNA repair capacity, so they are better in repairing their damage than the other tumor cells. Activation of the pro-survival pathway, so they activate different pathways which allow them to survive this redistribution on the different stages of the cell cycle. They have also very strong capacity to protect them against oxidative stress by different mechanisms. They also have unlimited self-renewal potential and impaired cell cycle. And they have extrinsic determinants such as hypoxic microenvironment and the protective cancer stem cell niche. So this all in the context of 5R makes them really tough to kill. So basically, they are one of the main reasons of the treatment failure, and we have to really consider it when applying radiation. So with this, I come to one of the last questions, which is, why are cancer stem cells considered as major challenge in radiotherapy? So again, there is only one correct answer. Yeah, I try to summarize it. So
Lisa, while they're answering, when you were showing your slides about niche and the cancer stem cells, I was thinking, isn't it contradictory, perivascular and hypoxic?
so yes, it is, but I think this is also, well, a choice of cancer stem cells to choose in which niche they are, and I think it also depends on the type of the tumor. So there are in general tumors which are more hypoxic and this way more resistant, like head and neck, for example, is more radioresistant than prostate cancer. This is the one which we're working with. So I think it really depends.
So it's or, okay.
I would say or, yes.
Or, okay, great.
I would
Thank you.
add-
So sorry, go ahead.
I would like just to add that it's not always like a mutually exclusive. There is also paradoxically niche found which is perivascular and hypoxic due to, for instance, in glioblastoma due to not fully functional microvessels. So it's sometimes it could be combined.
Oh, interesting. Okay, thank you. Thank you for clarification. So ending the poll, sharing results. So 80% answered B.
Very good, 80%. This is how it is. Yes, that indeed they have efficient DNA repair. They have protection against oxidative stress, and they can drive the tumor recurrence. So this is true. This is the cancer stem cells. The take-home message of this part is that cancer stem cells is a small subpopulation, but strong, unfortunately, of tumor cells, with unique capacities of self-renewal and ability to give rise to the diverse lineages of cells that comprise a tumor. They are therapy resistant, have high metastatic potential, and that traditional radiotherapy effectively kills the bulk, so other tumor cells, but not always the cancer stem cells. And the cancer stem cells are the one which, if not eradicated or not removed, they will eventually repopulate the tumor, leading to the clinical recurrence. So now we talked about different stages, and coming to the question of the tissue and cellular response. So we already looked on the molecular level, so here on the DNA level and the DNA damage, and what happens basically immediately inside the cell after radiation, and the tissue level is basically how the entire organ or system, like those in the images before, will behave over the time. So we will now touch this topic a bit. So as mentioned already, to refresh your memory, on the molecular level, when we look at the cell or when we apply radiation, it causes indirect or direct effect, damaging the DNA. This leads to the cell cycle arrest and basically the death of the cell. Now we will talk on the cellular level what happens. So when the cell has to die, yeah, and cannot repair its damage anymore, it's also a choice. So the radiation-induced cell death is a complex biological choice dictated by the cell type, the dose, and the oxygen level in the tissues. And there are many ways how cell can die, but there are two main ways related to the radiotherapy or irradiation, which is the mitotic catastrophe and apoptosis. And these mechanisms are primarily driven by DNA damage, so such as DNA double-strand breaks. So we're coming here to the end point. We apply the radiation, radiolysis of water, DNA damage, DNA double-strand break, cell decides to die. So I will touch mostly those two main types of cell death, although mitotic catastrophe by itself is not the cell death, but I will explain it in the next slides. So mitotic catastrophe, what we have there? It's one of the primary mechanisms of the cell death following ionizing radiation. So we damage the DNA, and then a cell with this damaged DNA bypasses the critical checkpoints of the cell cycle and attempts to divide anyway. And its mitotic catastrophe is having specific characteristics. You can see this immunohistochemical image of the cancer cells. You can maybe pick immediately what is wrong with these cells. You can see that this the cell loses cell cycle checkpoints, but also the cell becomes really big, and it has multiple nuclei or micronuclei. So this is a sign of the mitotic catastrophe. As I said before, mitotic catastrophe is not the cell death basically mechanism, but when the cell is like that, it cannot go back in its state. So this usually triggers a secondary death pathway, which in that case is usually apoptosis. So apoptosis is the programmed cell death. So basically cell suicide. So cell is damaged, cannot repair itself, and it just decides to kill itself because it's easier for the whole system like that than maintaining it. So this occurs rapidly following the radiation-induced damage. And unlike the mitotic catastrophe, which happens during the attempt to divide, apoptosis is active and energy requiring process, and it can occur at any stage of the cell cycle. And it's characterized by a specific sequence of cellular changes. So you can see here also a real image of the cell, which is undergoing apoptosis. So the cell first shrinks and chromatin condenses. Then it leads to the fragmentation of the nuclei and the membrane blabbing, and it forms the apoptotic bodies, and the cells basically dies. So those are the two main ways which you have to keep in mind when talking about radiation. But there are cases when other cell death types can happen. And I prepared also a slide about other cell death types, which can be necrosis. Also here you can see immunohistochemical image. And so this three phases for the cell death I will talk about are very different. So necrosis is very violent and passive form of cell death. So on this image, it characterizes by this increased part of the, I don't know if you see the difference. So this is more purple and this is more pink. By this eosinophilia, so this pinkness. And these areas are usually appear more pink because of the denaturated cytoplasmic proteins, which are bind more to this pink dye than to the purple dye. What it characterizes, it occurs primarily with the very high doses of radiation. And basically the cell loses its ability to regulate ions, swells with water, and just ruptures. So that's there is autophagy. This is very interesting. I would say my favorite, if you can pick a favorite here, is a program cell death suicide. When I say program, it means that the cell kind of chooses if to kill itself just to kill, or actually has a benefit from it. And that's why also it's argued a lot that actually autophagy is not the death mechanism, but survival mechanism. It's because the cell basically breaks down into its own damaged proteins and organelles to generate energy. Yeah, so it can use its own particles of the cell to maintain itself. And that's why it's called the pro-survival mechanism. So there are proteins called ATG, so autophagy-related proteins, which make the autophagy possible. And these ATGs, they cause the formation of the structure called autophagosome. Its structure is stained here with green. So it forms, these autophagosomes, they carry these chunk pieces of the cells to the part of the cell called lysosome, and it's being lysed. Yeah, so lysosome job is to digest basically, or break down these pieces. Last, also not exactly the death, but related to that, is called senescence. So we call it radiation-induced senescence, is a form of the permanent cell cycle arrest. So the cell is actually not dead, but it cannot do anything. So it cannot divide. It's metabolically active. It lives, but it cannot divide. Yeah, and this is also triggered by DNA damage. It's a state of permanent cell cycle arrest. These cells remain metabolically active and alive, kind of, but can never divide again. So this is the classical secretome, so beta-galactosidase is a marker of the senescence for the staining in the biology. Yeah, so basically, this DNA damage, it acts like a double-edged sword. Yeah, suppressing tumor by halting the damaged cells, but promoting tissue aging in this way. So now when we talked about the cell death on the cellular and molecular level, finally we can look at the tissue level. And the radiation response at the tissue level involves actually very complex and multi-stage damage to DNA and cells, leading to the different things like inflammation, stem cell depletion, and depending on the dose, to the acute or long-term reactions, or like acute or chronic reactions. So acute reactions to the tissue, they can occur in days to up to 3 months. And this is usually happened to the tissues with the high turnover rate, which means like skin, intestinal epithelium, bone marrow, the one which are actively dividing. And acute, in the case of the acute reactions, is failing to, the tissue is failing to regenerate, it's increasing the vascular permeability and immediate inflammatory response. And when we talk about the chronic reactions, which can take from months to even years, so it usually occurs to the slowly proliferating post-mitotic tissues like kidney, liver, heart, brain, muscles, and we can see the tissue remodeling, inflammation, and then in the end, the fibrosis of the tissue. So the take-home message of this part is that radiation-induced cell death is a very complex spectrum. So you have choice, ranging from apoptosis, so the programmed suicide, to mitotic catastrophe, which is division failure, to senescence, which are basically zombie state, I called it. Yeah, so you are there, but you don't divide. Or autophagy, which is the self-eating, survival or death. It really depends. And necrosis, which is an inflammatory rupture. This all dictated by the repair capacity of the cell and radiation dose. So acute tissue responses occur within days to weeks in rapidly dividing tissues due to the immediate depletion of the stem cell pool. And the chronic or late responses, they manifest months to years later in slowly dividing tissues as a result of the progressive vascular damage and permanent fibrosis. So I think I'm good on time. I'm coming to the conclusion. And what I wanted to say here is that so the biological hierarchy, which we went through today, from DNA to the chromosomes and to the cell, so to the nuclei, and then to the tissue, these are fundamental building blocks that ultimately shape, in the end, the whole organism. However, in the clinical and scientific context, it's really important for us to remember that we are not just looking at the sequence of nucleotides or the set of chromosomes, that we are analyzing life at the DNA level. And this way we gain insights necessary to provide better care to the living organism. So in the conclusion, I would like to say that these R's which I presented to you today are the tools we use in the clinical practice to widen the therapeutic index. So the therapeutic index, and I promise it's the last information you get from me today, is the safety margin between killing the tumor and sparing the normal cells, so the person. So this therapeutic index is the relationship between the probability of tumor control and the probability of normal tissue complication. So you can see here that the aim of all of us is here to make it more wider, so to kill the tumor but to spare normal tissues. So if you are interested in radiation biology, you can contact me, or you can further educate yourself. So there is beautiful books, there are many editions of it, Radiobiology for the radiobiologist, and or for radiologists. So it's amazing series of books. I suggest you to get one of those, and it's really nicely from the physics to biology, really described. Of course, use the PubMed for the latest peer review scientific studies. What I also would like to advertise here is this channel. It's called Imagine Microscopy. It's created by our Ukrainian colleague who is now located in Netherlands, Anna Mucha, with the colleagues, and they are doing beautiful videos about how things work. So far, it's only one, and you see there are only 3 followers there. I hope there will be more after today. So it's a YouTube channel. They have beautiful videos, how cells metastasize, about cancer stem cells, and so on. So I really suggest you to subscribe, follow, like, share, repost, yeah, and really educate yourself. So with this, I'm coming to acknowledgements. I would like to really thank the Help Ukraine group, of course, with endorsement of Astro. So to Anna, Natalia, and Natalka who organized this, I think it's really amazing event, and I truly enjoyed preparing the lecture today. And of course, last but not least, I wanted to say thank you to everybody who joined from Ukraine. I can imagine how challenging is that. So my mom is in Kiev also, so I'm in contact with her, and I know there are electricity problems and Russia problems. But I'm really grateful that you could make it today, and I hope it was useful. So thank you so much.
Great lecture. Thank you, Lisa. I already saw a lot of acknowledgements and the compliments to you, but we are not done yet. So there are still some questions.
Yes,
And
very nice.
I also saw a few questions added to the chat. So perhaps we can start from them. And we
Yes.
can continue from questions from audience. I already saw already a few. Okay, so let's start from the first question. Okay, the first question is from Oleg Yarmak, and okay, so let me translate. So basically, Oleg is asking, how do we consider in our radiobiological research the individual response of the patients to therapy? So basically, what is clinical translation of the things you described today, Lisa? How it can be used for personalization of radiation therapy?
Yes, it's very good question. I love it. So this is basically what Anna, who was just talking, what her group and well with me together is doing. Our group is called Biomarkers for Individualized Radiotherapy, and we truly doing our best to make it really individualized. Of course, it's challenging. It's challenging because of many reasons. Well, first, the one which I named today. Secondly, it's the bureaucratic reasons because, for example, we have now, so what we try to do in our lab is not to do only the biology, so not on the cells or not even mice, but also confirm what we see in patients, yeah? And with this, we try to assess if we are actually successful in the biological studies we are doing or not. And one of our recent findings, for example, we do this liquid biopsy, which is a very promising tool, a non-invasive tool. Yeah, so you just take the blood from the patient to catch cancer cells in the bloodstream. And based on our previous knowledge from the biological experiments, we apply it now to the blood of the patients to evaluate if the markers which we identified. By markers, I mean the molecules or proteins, which we can see that they differentiate the response to the therapy. So, for example, we have a marker which we measure in the bloodstream, and we can see it's very much upregulated after finishing the radiotherapy, but it was really low level in the beginning of radiotherapy, and this way, by measuring it constantly, we can predict the response of the patient to the radiotherapy course. So this is our ultimate goal, and this is what we want to do and how we want to approach this individualized treatment. Yeah, so we want to stratify patients and know how they will respond to therapy before applying the therapy. I don't know if I answered your question.
I would like also to to add something, but also advertise our future lectures, because the biological basis of fractionation and modeling of radiobiological response will be in more detail described in one of the upcoming lecture. Unfortunately, I don't have now in front of my eyes exact date, but it will be delivered by Stefan McMahon from UK. And please follow the schedule. Educate yourself about the future, the title of future lectures, about the agenda, and I believe that you will also learn a lot of practical things, how to translate the biological knowledge into the doses and into the clinical practice in more detail in upcoming lectures. But what Lisa said, I fully agree. We also have more questions, actually, many more.
Okay.
Okay, so Dmitro Senchuk asking, you have said that tumor cells are smart enough. What did you mean? Do they act in some way or just divide and survive?
Well, it was just a saying. Actually, I would say so. Our colleague, I loved how he said it. If the cancer cells would be smart, they would not kill the host. Yeah. So by saying smart, I think it was just mentioned in the specific context, but indeed, if they would be smart, they would not make the person die in the end from cancer. Yeah. So they are smart in some of their protective mechanism, and I think it also has to do with the evolution or this evolution hypothesis of the cancer stem cells. But no, they're definitely not smart in the context we imagine it.
I would use this perhaps, smart, maybe not appropriate word here, but I would say
Exactly.
opportunistic, what exactly we discussed during the previous lectures, right? So yeah, this opportunistic mechanisms of tumor cells and making them very much individualistic and behaving independently on the host organism. Okay, so Lesia Mitsak asking how can we apply this knowledge about cancer stem cells in the radiotherapy treatment and try to diminish the radioresistance?
Yeah, so I think this is very relevant to the first question I was asked today about this individualized radiotherapy. So we have to, what is our group is also doing, we are trying to find ways to find, for example, the markers which would detect the cancer stem cells specifically. Yeah, so we have projects now on head and neck on the specific marker. I don't know if I'm allowed to mention it, but where we combine radiation therapy with immunotherapy with the modified CAR T cells, and this CAR T cells they find on the tumor cells or this cancer stem cells and eradicate them, so kill them. And we saw that in mice, we saw that in cells. We are not in humans yet, but the goal is that the markers which we identified, that they would recognize specifically the cancer stem cells, mostly, not only the bulk tumor cells.
Great. So there are again a lot of acknowledgements. Okay, so I'm going to the next question from Vladislav Telichko. So hello, Vlad.
Hello,
The
Vlad.
question is, hi, Lisa. There are a couple of questions regarding cancer stem cells. Do cancer stem cells respond differently to photon versus proton radiotherapy? Is it correct that radiotherapy can sometimes unintentionally increase the proportion of cancer stem cells and potentially make tumors more aggressive? Thank you, Vlad.
Thank you, Vlad.
Insider question.
I invite you for lunch, Vlad, to discuss this. Very good questions. So the first one, photons versus protons, I don't know. I don't know because there is not so much done on the protons. Yeah, it's due to the different facts, but we don't know yet 100% or not even 100%, but to the same extent as with photons. Yeah, how they react with the cancer stem cells. In theory, the protons, they have a higher linear energy transfer, which means that they probably do more damage. So even for this tough and robust cancer stem cells, it's probably hard to repair. So we have hopes that protons this way are more effective. And this way they probably can help us to overcome this radioprotective mechanisms of cancer stem cells. But this is my assumption. I don't have now literature data to confirm this immediately. Yeah, I don't know, Anna, you want to add something or I go to the second question? Yeah, so then I see you nodding. So for the second question was, yes, that radiotherapy unintentionally increased the proportion of cancer cells. This is an amazing question. So we believe that, yes, radiotherapy primarily kills the bulk tumor. Yeah, this is the reason why the cells or the tumor can remain, can metastasize, and so on. If the cancer stem cells in this case will survive due to these niches, and it can be that their proportion will increase. Yeah, but this is one of the reasons why we use also fractionation, and one of the things that I think in the context of proton, probably it also would be better in terms of killing than in terms of unintentionally increase the proportion of cancer stem cells. So, yeah, like that.
So thank you very much. But basically with fractionation, you can actually even more push the selection, because you every time you select the strongest cells. Okay, the strongest, that is in brackets actually. So
yeah,
if you select at the end, actually you make such a tumor which will not be possible to eradicate. So kind of.
I mean,
Maybe
I would
this is
say
a bit
it's a hypothesis.
this is a bit naive question from me, because this is
No, it's
I
fine.
consider this as an evolutionary process. You select
Exactly.
basically.
That's why I also said that I think that's this recently, let's say, offered evolution model make completely sense. But it may be also because of the plasticity that the cells which were not cancer stem cells will they differentiate and become stem cells. That's also possible. I agree. And they kind of will learn newly how to be stem cells. But let's put it this way, that so far radiotherapy is one of the best tools for the local control. And I guess probably we don't have something better. Maybe protons, maybe.
Yeah, thank you for the answer, and more questions during the lunch. Yeah.
Agreed.
There are also, I see also Lesia Pederko. You would like to ask something, right? So you are just, like keeping
Yes.
for a long time.
Yes.
Mhm.
Yes, thank you, Lisa, so much for this great talk. Your lecture is very valuable for me. I'm wondering about the way of cell. You mentioned different ways, and as I know now, many talks about apoptotic cell death. Is it possible after radiotherapy that cell goes by this way, apoptotic cell death?
Yeah, so it's I think it, so there are papers. I am not really an expert, I don't know, to comment on that properly. So as I understand, this is the way of the cells to die when they kind of develop the resistance to the classical apoptosis. Correct me if I'm wrong.
Yes, it's like a different
Was
way
it?
when the cell choose this way by immune reaction, and it does like bombing the self,
Yeah.
just despair all of from inside. It's very interesting kind of cell death, and now so many talks about it and papers, and
Yeah,
I'm
that
wondering so much about this question. This
yeah,
is
so I think probably in the context of this R's, it makes sense that this would be a strategy for the cells to bypass this resistance, this apoptotic threshold. So I think it is possible, yes, but unfortunately I cannot say more about this.
Okay, thank you so much.
I also see, I don't see any more any questions from the audience. There are also some couple of questions left from the chat. Let me see. Okay. Olena Romaniuk asking, thank you very much for the interesting lecture. Could you please explain what time interval is required between fractions during hyperfractionation? So perhaps may not be quite related to Lisa's lecture, but
yeah, so so I cannot, as I said, as a clinician or as not a clinician, I cannot really probably comment it properly. So as I understand is the hyper, not hyper, right? It was hyperfractionation, which means that the dose will be delivered in the even smaller doses. Do I understand it correctly? So, and probably more often than once a day. So at least here in Germany, it's 5 times, I think, a week or something. It's delivered. So hyperfractionation would be twice a day, maybe, but I think it has to be really considered by the clinician or by the radiation oncologist how it's delivered. I think it depends on what cancer is it, what is the turnover there, how fast the cells are dividing, if it would make sense or not. I would answer this way, but unfortunately I cannot give you more information on that.
So perhaps Natalia and Natalka also would like to comment from your point of view. Okay, so the next question is, I understand this is very strange, so from Dmitro Sanchuk again, but are there any studies, if you know, kind of relation with sociology? Well, I think it's a bit interesting question.
Philosophical.
Organism is a society and cancer cell are individualists that act against and destroy it. Well, I think, well, it's a bit more philosophical question.
Okay.
So
Well,
perhaps we don't have time now to really discuss it.
yeah, but thank you. You were listening carefully, I can see that.
Thank you for great lectures, Kirilo Teluha. If oxygenated tumor cells more radiosensitive, it would have some reason under oxygen therapy as radio modification. I think this question again referred to the first lecture given by Professor Krause.
Mm-hmm. But I can also comment. So I think I said already about this oxygen enhancement ratio, and there are studies. Yes, so you can call it oxygenotherapy. Well, not giving directly oxygen to a person, but giving agents which makes cells more radiosensitive. So this would be like pre-treatment before radiotherapy. But there are also ways when with oxygen the later effects of radiotherapy are treated. So it is the ultimate radiosensitizer, oxygen, but how to use it in context of like, I don't know, giving mask with oxygen, I don't know if it would work, actually.
Yeah, so it was actually a treatment like we discussed it during our first lecture when patients were treated with hypobaric called like oxygen, and well, with some controversial outcome at the end.
Yeah.
Yeah, so I'm just going through all multiple thank you. Just give me 1 second. Nadia Honcharenko asking, could you please explain if there are any difference between proton and photon therapy in terms of cancer cell damage, or it's just a profit in conformity of plan and easier OAR sparring? So I think it's yeah. So
Okay,
yeah, the question is just to make it shorter, organ at risk.
Organ at risk, yes. So I mean, there are different differences. Sorry, for potato, between protons and photons. And yes, it's the profit of organ sparing, and it's of course important. But it's also so the biological effect of protons, as I said to you already previously, this is not very well studied, or because protons are fairly new in terms of radiotherapy. Secondly, it's very expensive machines, so the access to them has a very limited number of countries and people. So making also biological experiments with protons, for example, for us is very challenging because we have proton time in the beam bunker once in 3 months. So it's really hard for us to do the experiments there, especially if we are talking about living organisms like mice. So on the biological level, I cannot tell you how much is the difference. Of course, it's higher linear energy transfer, so it's more damage. And you could see this on one of my previous slides where I showed you the blue gamma H2X cell with the traces of radiotherapy beam you can then detect by the gamma H2X. But it is definitely a profit for the organs at risk. Yeah, so we really also for irradiating very small volumes. So now I think our colleagues are able to irradiate, I don't know, like millimeters of the tumor or something in the mouse brain. So you can imagine it's really small. So I think the precision is also very important. Yeah.
I would like also to add that many clinical trials which are currently ongoing cannot clearly indicate that there is a huge benefit in terms of the killing of tumor cells. However, definitely there are a lot of evidences that organ at risk can be more safe. Now, as biologists, of course, we are interested to understand differential mechanism which could be potentially activated by proton versus photon. And so far, there are no clear evidences, unfortunately, that a proton can activate certain signaling pathways which are I would like to say that which can provide their let's say clear biological benefit compared to photons. However, we also need to keep in mind that now very new delivery technique, like for instance flash radiotherapy, clearly indicates that so first of all, we can make a statement that flash radiotherapy may be nicely combined with immunotherapy and potentially also activate immune response. We also can make a statement that proton therapy could be more safe in combination, for instance, with cell-based immunotherapy because it could spare immune cells. And it may also be more efficiently activating host immune response against tumor. We also know that, of course, proton may induce more clustered DNA damage. And we also have evidences, again, from laboratory and from animal studies that even cancer stem cells, again, it's a bit referring to what Vlad asked, the question which Vlad asked previously, that potentially cancer stem cells can be less resistant to proton radiotherapy. However, we need to take this with precaution because all this study coming from in vitro and in vivo, however, there is no clear evidences in patients, and I mean, we still need to make or just to collect more information from clinical trials. And thank you very much, Natalka. Natalka add some information on the previous question regarding hyperfractionation for head and neck. So Natalka added that no less than 6 hours between fractions as general rule, and because the most important responses achieved within 6 hours. Dmitro Sanchuk asking, when we say radiosensitivity, we suppose tumors, but what about individual radiosensitivity? So if you talk about radiosensitivity of normal tissue, then please come to next lecture and we will talk about normal tissue radio response. And I think it would be a very nice continuation of what we discussed today. And if we have no other question from audience, please don't be shy, because I think it's quite interesting topic, and we have a great opportunity to discuss it a bit more today.
You can also find me on LinkedIn and ask.
So I hope that actually I read all the questions from the chat. If not, please raise your hand and ask. It's not the case. And maybe I can also ask you, Lisa, because, you know, as a moderator, maybe you could also comment, because according to what you said, one could really make a conclusion that, okay, once, you know, the most critical subpopulation of tumor cells eradicated, like cancer stem cells, then we basically don't care about other subpopulations, and, you know, so, but how could you comment? So, for instance, if we know that certain subpopulations of tumor cells more aggressive, do you think that it would be reasonable just to focus all therapy, all clinical efforts to eradicate this population, or in fact, any tumor cells could potentially be an origin of tumor growth?
Yeah, so I mean, of course, to eradicate the cancer stem cells is so there is a difference between a treatment and a cure. Yeah, so we can treat, we can see the shrinkage, we can see that patient is feeling good again and so on, but then the tumor comes back. So the goal isn't just to make it disappear, but actually to destroy the niche and to clear completely, so that not leaving, you know, the blueprint of the tumor. Yeah, of course. As I mentioned before, we also have circulating tumor cells, we have circulating DNA, so the tumor cells are not just sitting there, they send their people around to the society, so you can find them also later in the different organs, and this is the tricky part of it. And there is also the seed and soil hypothesis, yeah, so that the seed represents the cancer stem cells, so they can also go to the different organs and undergo EMT, and the soil is represent the local environment. Yeah, so there are also places where stem cells or cancer stem cells or tumor cells or metastatic cells like to go and sit and wait for their moment, for the good environmental conditions. So no, I don't think that all the therapies have be directed to the cancer stem cells. Of course, we have to kill the bulk tumor as well, because we know that there is this plasticity, and we saw it also in our studies when the cells can change their status upon different conditions. Yeah, so becoming from non-cancer to cancer stem cells and so on. Did I answer your question, Anna?
Definitely. So I believe, Natalka, this is also a question to you. So
Yeah, I think Natalka left.
central nervous system tumors in pediatric practice. In our clinic, we treat them with 4-6 hours between fractions.
Yeah, so Natalka left. I think she wrote that she has to leave earlier. So I don't know. I unfortunately cannot comment on the regime of the fractionation therapy, but yeah.
So perhaps we can keep this question for our next session.
For sure.
If Natalka is not here, because actually we are chatting now through the WhatsApp.
Okay.
So nevertheless, we can also, yes, Natalka is here.
Okay.
This is Natalka Suchowarska, not Natalka Kovalchuk. So we generally have 6 hours. We don't generally hyperfractionate unless we need to get the fractions in to avoid a big break, for example, Christmas or something like that. 4 to 6 hours, yes, that's also reasonable. We try to keep it to 6, but I don't think there's a clear rule on this at the moment. I hope that answers the question.
I believe so. Thank you very much. Thank you.
Can I add another comment though, please? Excellent presentation. I've really enjoyed all of them.
Thank you so much.
But I'm also hearing the questions that I think are just fabulous to see, especially in this lecture, because they're coming from clinicians with a lot of clinical experience, trying to relate what they're hearing to what they're seeing in the clinic. And what we see in the clinic is very messy. People are different. They come with different tumors. They come with different stages. And there's a lot of noise in that information. So in radiobiology, the academic side, we try and put some order in what we see. And we can't deliver everything at once because it's not possible. And even in this lecture series, we are only doing the very surface. So for all the questions that I'm seeing, please come to all the lectures, because it will reveal, it's a story that we have to start at the very beginning, and it will bloom, and you will see different aspects. It's an extremely complicated area. And I hope that by the end you will see that, in fact, what we're doing by thinking technology is going to answer all the questions. I think the radiobiology is the next big reveal to where our treatment is going. So it's really important to understand all these processes, because this is what will be happening over the next 20, 30, 40 years. So I think the questions are brilliant. Fabulous means everyone's listening and beginning to think in this way and understand. And thank you to all the lecturers. I think it's been really well explained. Please keep coming. There will be more.
Yes, absolutely.
I would also comment on your comment, Natalka. I think it would be very important, perhaps, to make this round discussion, with like a panel of some of our speakers all together during our very last session of this course to talk and to discuss half the knowledge we discussed during the course, during the lectures, can be translated to clinic.
Yes, this is what we will be doing.
Yes, exactly. And it would be also nice if maybe some questions, maybe not the same, but, like if our audience would prepare some questions, for this last session when we can discuss, all this like open translational questions. I think it would be very useful for the audience and actually for us, because we also need to learn and to have feedback as biologists. So yeah, I think it would be maybe the best design for our last session. Great.
So please, I would encourage people to send a well-formed question. So if everybody sent one question, there are 100 and something people here. So we would have something to choose from and combine them. That would be really, really useful. But you don't have to rush, because there is a lot more information to come and your question may be answered.
Sure. Great additions. Thank you very much. Thank you, Natalka, for joining and for commenting. Okay, with this, I think we are ready to finalize our today's session. Thank you very much, Lisa, for your nice talk. Thank you very much for coming today. Thank you all people for joining and for actually participating. It's really touching us. And we, of course, here we are kind of barely understand the truly situation in Ukraine, but we are, our heart and our soul definitely with you. And yeah, so we are very much looking forward for our next meetings.
Thank you so much. It was really a pleasure, and yes, looking forward to the next lectures.
So we will see each other in 1 week. Thank you very much, and talk to you soon.
Bye.